Gene prediction is the problem of locating the genes within the genome of any organism. As you know that in human there is only 3% genes are present in the genome. It means that only 3% of the human genome is coded into the proteins. The leftover 97% is so-called junk DNA. But now today we know that there is no such word or a terminology of junk DNA. More than 90% of the so-called junk DNA is also transcribed into a non-coding RNAs. And these non-coding RNAs, they perform very important functions in different biological processes. For example, in the brain development and in many important diseases, including the cancers. And up till now, there are very few in silico softwares that uses the approach of an algorithm to predict the presence of genes within the genomes of particular organism. And we also know that the split gene model or the exon-intron gene arrangement is only present within the eukaryotes. And it is very difficult to predict the genes in the eukaryotes. But on the other hand, in the prokaryotes, for example in bacteria, there is no such presence of introns in the genomes of these bacteria. So it is much easier and much simpler to find the presence of genes within the genome of the bacteria because there is no introns in the genomes of bacteria. So to predict the location of genes within the genomes, the scientists, they use two main strategies. Number one is the statistical approach, and in the statistical approach of the gene prediction, the scientists, they focus on the very specialized regions of the genomes that are very conserved within the genes. And one of these conserved regions, they are called the splicing signals that are present in the exon-intron junctions. And the example of these splicing signaling is the presence of A, G and GT on the left and right side of the exons. And these A, G and GT, they are highly conserved at the junctions of the exon and introns. We can see here the splicing signals are present in the junctions of the exons and introns. Here we can see this is exon number 1, 2 and 3. If there are 3 exons, there must be 2 introns. And this is the junction of exon and intron. And as we have seen that before the start of the intron, there must be present GT, and at the end of the intron, AG, GT, AG, and so on. Whenever there is an intron within the exon, it will start with GT and ends with the AG. So we can now predict the presence of the exons in the gene, and by using this information, if it is GT and AG, we can predict that this is the start of intron and end of intron, and definitely before start of intron there must be exon, and after the end of intron again exon. So we can predict the presence of exons within the gene. So the less conserved position on both sides of the exons, the simplest way to represent such binding site is by profiling, describing the properties of different nucleotides to occur at different positions. We have seen that we can use the splicing information that is present at the junctions of introns and exons, and by using profiles to detect splice sites has met with limited success, since these profiles are quite weak and tend to match frequently in the genomes at non-splice sites. Because we know that GT and AG, it could be present at any position within the genome. So it could be a misleading information when we were using that statistical approach, and it can match with any of the other sites than the introns. So the attempts to improve the accuracy of gene prediction led to the second category of approach, because we have seen that this is the first category of approach, that was the statistical approach. And in this approach, scientists, they mainly focus on conserved regions or conserved parts of the gene. And the other approach is the similarity based approach.